Okay, in the last one we talked about the different kinds of mutations and different kinds of mutations and how they can kind of alter the phenotype. In this one we're going to look at mutagens and the phenotypic effects of mutations. So let's get into it. Mutagens are substances that increase the mutation rate during DNA replication. We have chemical mutagens, such as base analogues that alter base pairing, intercalating agents that lead to insertions or deletions, or chemical modification of bases, for example, through deamination, the removal of amino groups. You also have physical mutagens, like ionizing radiation, like X-rays and gamma radiation that break DNA strands. And you also have non-ionizing radiation, like UV light, that causes bases to become dimers. An example of a famous chemical mutagen would be ethidium bromide, which, if you do biology or any kind of genetics work, or you see it in videos, we use ethidium bromide when we're running gel electrophoresis, because it kind of binds to the DNA, making it a bit more visible. We add the ethidium bromide in the ionic solution that we place over the gel when we're running our samples. So, going a bit into, we talked a bit about the transcription process, and then the translation. The ribosomes converting codons into polypeptides. We're going to talk about that in a bit more depth now. So, transcription, basically, you get your double-stranded DNA in the nucleus. A DNA helicase breaks the hydrogen bonds, opening the double-stranded DNA. You then get a DNA polymerase, which copies. It uses the DNA as a template strand, produces a strand of pre-mRNA. This pre-mRNA then undergoes a process called splicing, where a spliceosome cuts the pre-mRNA at the intron-exon junctions, removing introns, and then a ligand will bind exons together to produce a molecule of post-mRNA, which will then travel to the ribosome, where the codons of that post-mRNA are going to be read, and they're going to produce a polypeptide with amino acids put in the sequence that the post-mRNA is coded for. So, a modified GTP cap is added to the 5' end of the mRNA, and a poly-A tail is added to the 3' end of the mRNA. This GTP cap regulates the stability of the mRNA tubule and aids in recognition at the ribosome, while the poly-A tail also regulates stability of the mRNA, as well as preventing enzymatic breakdown of the structure and aiding in recognition at the ribosome for translation. It also plays an important role in the transport of the post-mRNA out of the nucleus. So, getting into the phenotypic effects of mutation, some mutations that we test for in a classroom are caused by altered structures of olfactory receptors, where they become unable to pick up their target molecule, so are unable to stimulate an action potential for a certain scent, resulting in what we call an anosmia. So, like we talked about here, these different kinds of mutations, they can result in the structure of the polypeptide and the protein once that polypeptide is folded. If the polypeptide has different amino acids in it, the protein is going to have a different structure. And if it has a structure that is no longer complementary to the molecule it's detecting, in the case of a receptor, then the receptor can't pick up its target molecule, so it's basically useless. And if we ever come into contact with that target molecule, you're not going to have the kind of the response that we would usually have caused by that receptor, because the receptor has basically become dysfunctional because it's the wrong shape caused by mutation. The most famous kind of anosmia you probably heard of comes from the scent of urine after eating asparagus. So it comes from a receptor that's responsible for picking up a chemical called asparagusic acid, which comes from eating asparagus. Those unable to detect the asparagusic acid in the urine have an anosmia caused by a dysfunctional olfactory receptor for this chemical. So our asparagusic acid receptor has a mutation in it. The mutation results in a different amino acid being incorporated into the polypeptide. This results in the polypeptide being folded at the Golgi apparatus into a different shape. So the receptor is a different shape. This different shaped receptor is unable to pick up the asparagusic acid and trigger the action potential responsible for us getting the smell of the chemical. So if we ever come into contact with it and we have a mutation that affects this receptor, we can't smell the chemical, so we have an anosmia. And these are basically the common kind of mutations that we look at in the classroom because they're harmless. So there's no real harm in us having a look if we have this kind of mutation. It's just an easy way to determine if you have a mutation.